And today we'll be discussing one of the most pressing challenges in modern medicine, which is antibiotic resistant superbugs. And these are the bacteria that have evolved to withstand the medications we have traditionally used to fight them. So, the title of the Insight into Science talk for today is The Chemistry Behind Beating Superbugs, the Global Challenge. Antibiotic resistant superbugs, as I mentioned before, these are bacteria that have developed resistance to the traditional types of antibiotics that have been used to fight bacterial infections. So, let's first just understand what these superbugs are. Looking at this diagram on the left, we can see how natural selection works in a bacterial population. Initially, we start with a population where most of the bacteria are susceptible to our antibiotics. However, when antibiotics are introduced and, in fact, overused, the resistant bacteria survive. The other bacteria do not survive, and these resistant bacteria multiply, eventually becoming the dominant population and therefore a problem for us. As we can see, this is evolution in action happening right before our eyes. As per the World Health Organization, what causes these antibiotic resistance? They have identified 6 key factors behind antibiotic resistance, which is overprescribing of antibiotics by healthcare providers, patients not finishing their treatment and their prescribed antibiotic doses, overuse of antibiotics in livestock and fish farming, poor infection control practices in hospitals and clinics, lack of hygiene and sanitation, and the most important one being lack of new antibiotics being developed. While there has been significant progress in the discovery and development of new form of antibiotics, they're still not in parallel with the antibiotic resistance, which evolves very quickly. So, understanding these causes of antibiotic resistance is crucial. Why? Because antibiotic resistance threatens to undo decades of the medical progress that we have had. Each of us has a role in preventing the spread of these superbugs, from proper hygiene to responsible usage and avoiding the overusage of antibiotics. Just to highlight the scale of the problem. So I have a question for all of you. And if you would like, you could please type on the answer you think in the chat. Which of these common infections do you think can now be caused by antibiotic resistant superbugs? A. Urinary tract infections, B. Pneumonia, C. Skin infections, and D. All of the above. I'll just give you 1 minute here to type in the answers in the chat if you would like. And while you think upon that, and I can see many people have already answered, let's look at the deaths that occur based on the drug resistant infections. As of now, we can see that there are multiple diseases such as tetanus, measles, road traffic accidents, diarrhea, diabetes, and the cancer being the most prominent with 10 million annual deaths in 2020. But as per the UN Environmental Programme and the World Health Organization, by 2050, this figure of antimicrobial resistant infections causing mortality about 1.3 million annual deaths will increase up to 10 million annual deaths by 2050, which is a big figure. And as you can see, comparable to cancer. And we're all scared of cancer. So now we need to be scared of these superbugs. And as most of you have said in the chat, as I can see, the answer is all of the above, which is quite scary because there are these multiple type of bacterial infections that can now be caused by superbugs that we don't have the medicines for. So, in order to understand the antibiotic resistance problem, we need to understand antibiotics. So, antibiotics are medicines that either kill bacteria or stop them from growing, and they are thus used to treat, cure, or prevent bacterial infections. So, these antibiotics, they can be classified based on different mechanisms. First, we have the spectrum of activity. So, based on this spectrum of activity, they can be divided into being broad spectrum antibiotics or narrow spectrum antibiotics. Broad spectrum antibiotics can target many types of bacteria, while narrow spectrum antibiotics focus on specific types of bacteria. Second, by their mechanism of action, how they actually fight the bacteria and the infection. Then the important one being type of action, which is either they're bacteriostatic or bactericidal. Bacteriostatic means they essentially just reduce the growth of the bacteria, but bactericidal antibiotics are essentially killing the bacteria, leading to cell death and henceforth. So, beta lactam antibiotics is going to be the primary focus of our talk here. How do beta lactam antibiotics work? While there are several classes of antibiotics, as mentioned in the previous slide, that can work in different ways, beta lactams are the most clinically important class of antibiotics till date. So, another question for you here, and you can type that in the chat. What does a beta lactam mean? Is it a four-membered cyclic ester, a four-membered cyclic amide, a five-membered cyclic ester, or a five-membered cyclic amide? It's a technical chemistry question, and I'm happy to look at. I'll give you 1 minute here to type up your answers. Okay. Um, So a beta lactam essentially means a four-membered cyclic amide, and we will see the structure of this molecule pretty soon. Another question I would interested in, if it's somebody would like to answer it in the chat, is what is the first beta lactam, or in general the first pure antibiotic that was discovered by Sir Alexander Fleming in 1928, known as? That's correct. You got it. It's called penicillin. And the answer for the first question can be explained through the structure as well. So here we have the general structure of penicillin, which is beta-lactam core structure molecule highlighted in green here. So this is a four-membered cyclic amide. So how do beta-lactam antibiotics such as penicillin work? These antibiotics are not bacteriostatic. They do not reduce the growth of bacteria. In fact, they are bactericidal. That is, they work by killing the bacteria. They bind to specific special enzymes that bacteria produce, which are known as penicillin binding proteins, via the core beta-lactam ring. Thereby, they interrupt the bacterial cell wall formation, and thus the bacterial cell dies. Here is a video for you on penicillin.
This video is all about the antibiotic drug penicillin. Alexander Fleming worked at St Mary's Hospital in London as a bacteriologist, and in September 1928 he returned from his summer holiday to find that his petri dishes had been contaminated from some mold from food. After examining these samples under the microscope, he realized that the mold had prevented the bacteria from growing. We now know that the chemical responsible for this was penicillin, which went on to become the first ever antibiotic drug. But how does it work? Surrounding bacterial cell membranes are cell walls made of peptidoglycan. The structural integrity of these are maintained by transpeptidase enzymes. These enzymes connect the sugar molecules together via short peptide molecules, thus forming the peptidoglycan. This bacterial cell wall is the target at which penicillin exerts its antibiotic effects. In gram-positive bacteria, penicillin can access the cell wall and membrane directly, but in gram-negative bacteria, it enters via transport molecules known as porins. Once within the bacteria, it binds to the membrane protein, imaginatively named penicillin-binding protein. This activates the penicillin molecule, allowing it to dissociate and bind to transpeptidase enzymes. This inhibits the enzymes, preventing them from maintaining the structural integrity of the bacterial cell walls, which eventually break down. This inconsistent cell wall causes water to flow into the bacterium uncontrollably as it cannot maintain its osmotic gradient. This quickly causes cell lysis and death. The discovery
Yeah, so as explained in this video, the mechanism by which beta-lactam antibiotics, such as penicillin, have a very fascinating mechanism. They contain this specific core structure of a beta-lactam, which binds to special proteins called PBPs, which are very important for the bacterial cell wall formation. So, how does bacteria develop resistance to these very interesting antibiotics? So, there are different mechanisms bacteria have evolved in a way to develop clever ways to resist these antibiotics. This diagram shows us five main mechanisms. First, we have the efflux pumps. So, some bacteria develop efflux pumps that basically act like tiny cellular ejection systems that pump the antibiotics out before they can do their work and have the bactericidal effect they would like. Second, decreased uptake, which is the permeability of the bacterial cell wall. So bacteria can develop mutations in a way that the permeability of its cell membranes and cell walls decreases. Therefore, the antibiotic dosage in the bacterial cytoplasm decreases. Next, we have modified target. So this essentially means that the penicillin binding proteins, which are the target for beta lactam antibiotics, can have several mutations. Thus, the antibiotic does not reach its target or does not do the desired reaction with the target as we would like. Fourth, we have the target overproduced by the bacteria. If the bacteria mutates in a way that that the penicillin binding proteins are overproduced, so the dosage of the antibiotic prescribed does not match with the target, we have resistance in the bacteria. The most important one here being inactivating enzymes. So the bacteria can basically produce certain enzymes which can inactivate our antibiotic. They can either degrade the antibiotic or attach a specific molecule to the antibiotic which inactivates it towards its future use for the reaction with penicillin binding proteins. And that is the focus here. So the key enemy for us is beta lactamase enzymes. It is the biggest challenge in fighting antibiotic resistance. These are remarkable defense weapons that bacteria have produced to provide resistance against beta lactam antibiotics like penicillin. These inactivating enzymes basically function by breaking down the beta lactam ring of the antibiotic. As you can see in this molecular diagram, these enzymes specifically target the beta lactam ring, the core structure that makes these antibiotics effective. When beta lactamase encounters an antibiotic, it breaks down this critical beta lactam structure, transforming the antibiotic from its active form to its inactive form, which has its beta lactam ring hydrolyzed. Think of it as the bacteria having molecular scissors, which cut up our antibiotics before they can do their job. That is, react with the penicillin binding proteins, hinder the cell wall formation, and causing the cell death. So these enzymes have evolved over time. There are four major classes of beta lactamases: A, B, C, and D. However, we have made new beta lactam antibiotics which are resistant to certain types of beta lactamase strains, but beta lactamases have been evolving even faster than the drug discovery and development process that has been going on. So, what do we do? Do we give up with all this evolution? Of course not. We have not given up this fight. One of our most crucial weapons against resistant bacteria is our beta lactamase inhibitors. On the left, we have a bacterial cell without a beta lactamase inhibitor. What happens here? We have the beta lactamase, which essentially degrades the beta lactam antibiotic, which cannot thus bind with the penicillin binding proteins. And these penicillin binding proteins are thus free to make a stable cell wall and keep the bacteria surviving alive. While on the right side, we have the addition of a beta lactamase inhibitor. So these are specific chemical molecules which can react and inhibit the function of beta lactamase enzymes, which are our enemies. So these beta lactamase inhibitors bind and deactivate the beta lactamase enzyme. Then the beta lactamase enzyme cannot break down the beta lactam antibiotic. So the beta lactam antibiotic binds with the penicillin binding protein, interrupts the cell wall synthesis, and eventually kills the bacteria. This is why, in the current modern world, we often prescribe antibiotics combined with a beta lactamase inhibitor. It's like sending in the troops with a shield against the bacteria's defenses. So, let's dive deeper into how beta lactamases actually destroy our beta lactam antibiotics. How do they destroy this core beta lactam structure? This mechanism is very fundamental and fascinating, and understanding it is very crucial for developing better treatments in the form of antibiotics as well as beta lactamase inhibitors. So we have a beta lactam antibiotic with its core beta lactam structure. And for the beta lactamase enzyme, there is an active site residue which usually contains a nucleophilic a hydroxyl group. There are two main types of beta lactamases, serine beta lactamases and metallobeta lactamases. We don't need to go into the details of them, but the main difference is what is the form of the active site residue in those enzymes. So in this case, the hydroxyl lone pair, or sometimes as an anion, acts onto this electrophilic carbonyl center where the electrons shift towards the oxygen and form a tetrahedral, a high energy tetrahedral intermediate. This is a temporary molecular structure. So after that, the negative electron density on the oxygen comes back and the fission of the key beta lactam bond occurs, and the nearby base in the enzyme helps in this process because this allows the amine to be protonated, and we then get the acyl enzyme complex with the beta lactamase enzyme. At the later stage, finally, water comes into the process, resulting in a hydrolyzed inactive antibiotic. This molecule is not an antibiotic. It does not react with penicillin binding proteins, and thus beta lactamases have done their job. Another thing to think about is that it looks like a multi-step reaction, but beta lactamases are regenerated at the last step, which means they can go again to react with another antibiotic. So essentially, it takes beta lactamases less than seconds to destroy all of the antibiotics that are functional. So a key question for you, it's a bit of a technical question. Happy to give you some time to think about it. What is the key driving factor for such hydrolysis of these beta lactams? Is it the electrophilic nature of the carbonyl bond to which the nucleophilic residue of the beta lactamase acts? Is it the formation of a high energy tetrahedral intermediate? Or is it the ring strain release that happens due to the breaking of the four membered lactam ring? Just give you 2 minutes to think about it here. Have a look at the mechanism and just have a thought. No answer is wrong here. So happy to receive any answers. I'm glad to see people are giving answers. Good. Okay, so very impressive here. So that essentially explains it. None of them are wrong here. All of those factors are very important in this process, and all of those factors are responsible for the hydrolysis of beta lactams. But as I mentioned, the key driving factor is C, which is ring strain release for the four-membered lactam ring. And it's only a matter of 10 to 20% difference of the other factors not being that impactful. And thus, this is the main reason why most of the antibiotics are beta lactams, because this four-membered lactam ring, when released, releases a lot of energy, and that is the key driving factor for this reaction. That's why the drug discovery processes and pharmaceutical companies and academic environments have been focused on only this beta lactam structure. Whenever they have tried to drift away from it, they have struggled to get the antibiotic efficacy that they would like. So, how can this mechanistic insight be useful in designing beta lactamase inhibitors, which are our crucial weapons for destroying beta lactamases? So, if we look at this mechanism, we have a nucleophile reacting with an electrophile. As simple as that. Then we get a high energy tetrahedral intermediate, and we have an acyl enzyme complex which is formed. So what we can do is, we know that beta lactamase enzymes have certain nucleophiles which are very reactive. So what we can do is synthesize new chemical compounds which can react with beta lactamase enzymes before they react with the beta lactam antibiotic. So, this mechanism allows us to identify new chemical compounds which are capable of undergoing a similar covalent reaction with beta lactamases. We can essentially create molecular decoys that keep these enzymes busy and away from our antibiotics. So, currently, we have two main categories of beta lactamase inhibitors in clinical use. First, we have the beta lactam based inhibitors, which are clavulanic acid, sulbactam, and tazobactam. As you can see highlighted in green, even these molecules have a core beta lactam structure because that allows them to react with beta lactamases due to the driving force being the ring strain release. And these beta lactam based inhibitors are known as suicide type irreversible inhibitors. They react with beta lactamases irreversibly and thus have the antibiotic destroying the bacteria. The other type of beta lactamase inhibitors, which are not conventional but have come up over time, are some compounds known as urea derivatives. So the example here shown is avibactam and relibactam. These molecules here, as highlighted in yellow, do not possess a beta lactam core structure. But the key thing to note here is that both of them have the core carbonyl structure, which allows them to act as an electrophile, which allows the beta lactamase nucleophilic group to react with the carbonyl group. And another interesting molecule, which is in clinical use towards being a beta lactamase inhibitor, is known as Weberbactam. These molecules are especially very interesting because they do not have the traditional beta lactamase, beta lactam, or any carbonyl bond within them. But what's interesting about these molecules is that they have boron. So, if in the chat anybody could mention what is the hybridization of boron in this structure? Is it sp, sp2, sp3? Okay, so the hybridization of boron in this molecule, because it has three bonds here, is sp2. So a special unique ability boron possesses is its ability to transition between sp2 and sp3 hybridization. So when the nucleophilic hydroxyl group of the beta lactamase enzymes react with such molecules, what happens? They react with the electrophilic center, which is boron, and then makes this into a sp3 hybridization. So I think I've answered this question for ourselves. What are the functional groups highlighted in yellow and blue called? As I said before, these are urea and a boronic ester. And the key important factor here being that we are synthesizing new electrophiles, new electrophilic type of chemical compounds which can act as molecular decoys, react with beta lactamase enzymes, either form a tetrahedral intermediate or form an acyl enzyme complex, which mimics them as the core beta lactam antibiotic. So I think I'd just like to highlight here my PhD research. During my PhD, Professor Chris Schofield at University of Oxford, I was working on designing and developing new templates of beta lactamase inhibitor. Just a highlight of something that I told you about in the last slide. So these molecules here are also boronic esters. Just difference being this is a 5-membered ring as compared to the previous slide, which is a 6-membered ring, and it's fused with a benzene ring here. So I was synthesizing C6 N substituted benzoxaborols, which could react with nucleophilic hydroxyl group of beta lactamase enzymes and form reversibly these enzyme benzoxaborole complexes, which are tetrahedral complexes. And what do you see? These tetrahedral complexes are high energy tetrahedral intermediates. They mimic the tetrahedral intermediates that are formed by beta lactam antibiotics when they react with beta lactamase enzymes. So during my PhD, I was varying the group R here and just a question here for everybody to put in the chat. What can be different R groups here? What functional groups can an amine react with? So can an amine react with a halogen? Can an amine react with a carboxylic acid? Can it react with a sulfonic acid? Anything that comes up in your mind. Anything else? That's right, that's great. So we have the usual carboxylic acids. So when carboxylic acids react with amine, they form amides. When sulfonic acids or sulfonyl chlorides react with amine, they form sulfonamides. When we have aldehyde reacting with an amine, we get a CH2 amine, a different kind of amine, alpha hydroxyl amine. So we can get different kinds of substitutions. And that was essentially my PhD. I was making different amides, sulfonamides. We could also make ureas from here by reaction with an isocyanate molecule. So we were trying to make different molecules which could then react with beta lactamase enzymes and have potency towards beta lactam antibiotics. The second part of my PhD research was a 5,5 fused thiophene gamma lactam. So it's essentially a gamma lactam and not a beta lactam because it's a 5-membered ring. And this gamma lactam was fused with the thiophene ring. And similarly, as beta lactams, the carbonyl bond being electrophilic here, these beta lactamase nucleophilic hydroxyl groups can react at the electrophilic carbonyl center, which can then give rise to an acyl enzyme complex, very similar to the ones that beta lactam antibiotics form. So, just a question for you. We discussed the driving force for the reaction for beta lactam antibiotics was the ring strain release of the 4-membered beta lactam ring. What do you think? Is the ring strain release for gamma lactam less or more than a beta lactam? Essentially, is the gamma lactam cyclic bond stabler than a beta lactam bond? Which one is more stable? Yeah, thank you, Alfie. Yeah, so it's less stable. So gamma lactam bonds are more stable as compared to beta lactam bonds. The energy release due to the ring strain release is less as compared to beta lactam bonds because they are 5-membered rings instead of 4. So that explains why I have drawn a reversible reaction here as compared to the beta lactam reaction, which was irreversible. And as we have seen previously, these beta lactamase inhibitors are irreversible suicide type inhibitors. They essentially sacrifice themselves to react with beta lactamase enzymes, while these boron based compounds known as benzoxaborols, due to their ability to transition between sp2 and sp3 hybridization, and the gamma lactam based compounds, due to their ability to have less string release, can form reversible type inhibitors. And how is this essential? How is this useful to us? When they are reversible, that can keep the enzyme busy over a long period of time, and they have less side effects. They do not interact with other bodily enzymes which are essential for the gut or for the human body. So, just to summarize, we have this immensely important fight against superbugs. What are the promising solutions that are currently in development? We have new antibiotic beta lactamase inhibitor combinations. Similar to as my research, there are different types of new chemical scaffolds that are coming, that people are developing towards being beta lactamase inhibitors, which can eventually be used into clinic. Novel drug delivery methods, because we have these big and drugs looking like drug molecules which essentially do not reach the target enzyme, so we can have novel drug delivery methods. Structure guided drug design is very important in today's world. So we have a new molecule and we want to see if it does the job that we want it to do, which is react with the beta lactamase enzyme. I showed a niche active site residue for the beta lactamase enzyme. That's not how it is. Beta lactamase enzymes have hundreds, thousands of amino acids. So, what other amino acid groups can react with these and with these chemical compounds? So, all of that can be essential if we use structure guided drug design. If what group, for example, on the sulfonamide group or the N nitrogen group, what group can be changed so that it effectively interacts with these enzymes in a better way? Alternative approaches, while where the main current focus has been actually trying to form new antibiotics which are not beta lactam based antibiotics. Not many have been successful in this attempt, but it's currently going on. And what is the global response that we should be looking at? We are looking at, which is worldwide research collaboration. There is investment in new drug development. And as not scientists, we should focus on prevention of these bacterial infections by sanitary measures and promote using antibiotics wisely. Do not leave your antibiotic dosage unfinished, etc. But chemistry, as I said, is our strongest weapon in this fight. By understanding how bacteria resist antibiotics at the molecular level, which is what beta lactamase enzymes being the key factor for the different types of resistance mechanisms, we can design better drugs to overcome resistance and save lives. I think World Health Organization says it better than anybody, which is the future of antibiotics